Hello everyone, my name is Sucheer Maddury and I'm presenting this talk from San Jose, California. Now, my presentation is titled AutoHD, which is an algorithmic biomedical signal processing technique for Huntington's disease prognosis. So as a brief background, the reason I actually embarked on this project was to further explore interdisciplinary machine learning as well as signal processing. So before we get to the bulk of the project, let's begin on Huntington's disease. What is it and why do we care? Essentially, Huntington's disease is a genetically determined neurodegenerative disease, which essentially means that its genetic cause is present since birth. However, the symptoms only set in at around the middle ages, such as the 40s or 50s, and it affects hundreds of thousands of people across the world. Now, the genetic cause itself is actually cytosine, adenine, guanine repeats in the HTT or Huntington gene, which is essentially causes protein misfolding in certain neurons in the brain, and that's what causes the neurodegeneration. Specifically, those regions are the basal ganglia and cerebral cortex. Now, these regions of the brain are responsible for functions such as behavior and motor control. And as a result of this, that's why we say that Huntington's disease is characterized by its motor, cognitive, and behavioral disturbances. With that out of the way, let's get to why we even need a new early prognosis method. Don't our current methods already work? Well, here we can see this chart, which was created by other researchers. And it shows that most people today are diagnosed in the early symptomatic stages of the disease. However, if we were to prognose them much earlier, before any symptoms had set in, we'd be able to use some sorts of novel treatments, such as gene therapy and deep brain stimulation, and that could improve the quality of life of the patient. However, the only real early intervention methods right now are things such as genetic testing, and these are extremely expensive, which is why no one gets them. So that's why we need new methods that are less expensive, but still reliable. That's where biomedical signals come in. Biomedical signals are essentially a way of tracking some value in the body over a continuous period of time. I used three in this project. The first is called electroencephalography, or EEG. Essentially what EEG does is it records electrical activity in the brain through a web of electrodes. There's also electrocardiography, which does the same thing except for the heart. Finally, there's functional near-infrared spectroscopy, or FNIRS, and this records blood flow in the prefrontal cortex region in the brain. And the important thing to understand here is that these abnormalities in the body actually set in before any symptoms, which is why it's plausible that we could use it in order to diagnose the disease before any symptoms set in. The objective of this project as a whole was to evaluate the efficacy of biomedical signaling as a biomarker for Huntington's disease. And the approach I settled on is to first extract features from the data and then apply some sorts of algorithms to predict Huntington's disease. The question I wanted to answer was, can biomedical signals expose abnormalities caused by Huntington's? So let's look at the data I used. The data I used was curated very recently, and it was conducted in a neurological clinic in Slovenia. I had a brief talk with the researchers, and they allowed me to use their data. It included 69 patients, 27 of which had Huntington's disease, 43 did not. There's no stimuli given to the subjects, which essentially means we're just recording typical brain activity and other activity as well. And we acquired EEG, ECG, and FNIRS data in 20-minute samples. On the bottom, you can see some attributes about the data, such as the number of electrodes or the frequency. Now, before we apply any sort of feature extraction, we need to get the signal ready. So that's what preprocessing is. If you take the EEG signal, for example, I first applied the 1020 reference, which essentially means I mapped the locations of the electrodes to the standard 1020 system. I then applied a FIR bandpass, which filters the frequency of the signal to some domain in order to eliminate the noise seen in high or low frequencies. Finally, I segmented the signal into 5-second segments, or epochs, with a small overlap, in order to create bite-sized chunks that the computer and model could more easily read. I did a similar procedure for the other two types of signals. Now, the specific reasons I used those domains were because the domain on which you only get the useful data with no noise just varies based on the type of signal. Now let's look at two important signal transform methods before we look at feature extraction. The first you may have heard of, and it's called the fast Fourier transform, or FFT. Essentially what the FFT does is it takes this complex signal and it breaks it down into standard sinusoidal waves with varying frequencies and amplitudes. And this way we can take a complex signal that we couldn't really understand and break it down into a bunch of waves that we do understand. There's also the wavelet transform. What this does is it takes this signal and applies a series of more complex oscillatory functions and maps the correlation between the signal and the function between each of them. And why is this useful? Well, for a signal such as ECG, the actual signal remains constant, but then there's these wide fluctuations. So wavelet transform could be useful. This is a list of all of the features I extracted. Now that's a crazy table over there. We can pretty much break it down into two types of features. The first is called the time domain features. And those are on the left. And you probably recognize many of them. Many of them are in your standard statistics or calculus class. However, there's also Hjorth parameters, which you may not recognize. These relate to the dynamicity of the signal. I also extracted frequency domain features. So these are the wavelet transform and fast Fourier transform signals. The wavelet features were mean, standard deviation, energy, and entropy. And for the FFT signal, I extracted this value called PSD, or power spectral density. This relates to the power of the signal over the frequency domain. I also constructed a robust model evaluation process in order to fairly represent the real-world model performance if it was applied in an actual hospital. I evaluated it on an epoch-by-epoch basis, so we're predicting Huntington's disease per segment. And in order to keep the segments together and avoid artificially inflating the accuracy of our model, I used a technique called cross-validation based on a group array, and I scored it on a variety of metrics shown on the right. So what happened after I did this? Well, these are the results that I saw. The ERT, or extremely randomized trees, performed the best, with random forest close in second. So based on this, I wanted to scale out the extremely randomized trees to see if we could get the accuracy even better through more estimators and a better loss function. I also scaled out the random forest since it was close behind. And this makes sense since extremely randomized trees are just a variant of the random forest model. So what happened after I did that? Did the model improve? Well, these are the results that I got. As you can see, the extremely randomized trees experienced about a 1% jump, and the other metrics improved as well, such as the F1 score. Now, these are the PRC and receiver operator characteristic scores. Now, I won't go into too much detail as to what these are, but they're essentially based on the true positive, true negative, and false negative rates of the model. And we draw this curve. We want the area under the curve to be as close to 1 as possible. But anything at 0.95 or above is considered good enough for medical diagnostic devices. So we can say that we achieved that here. In terms of statistical significance, if you did some math based on the number of electrodes, number of features, you'll get around 950 different features per epoch. About 580 of them were statistically significant with p less than 0.05. Now let's look at also which types of signals the model liked the most, that it factored the most into its decision. Now, there are a lot of charts here, but there are two important observations. The first is the significance of FNIRS, since it was almost as important as the EEG signal. And FNIRS was only invented a couple of years ago. So we want to further explore FNIRS through other diseases or explore the abnormalities in Huntington's disease itself. Additionally, the importance of the theta band in the EEG power spectral density was quite surprising since it's sandwiched between two much more important signals. So we can say that there is maybe some noise in this signal. Finally, what's the significance of all this? Why does it all matter? Well, with the auto HD procedure, we can now analyze lengthy biomedical signals in tandem that individual doctors would never be able to do by hand. We can also track abnormalities in the electricity of the brain and blood flow, and thus track the severity of the disease over time. And of course, we can diagnose Huntington's disease accurately before symptoms set in, which qualifies as early intervention. So what's the takeaway of all this? Well, AutoHD has shown that biomedical signals are a useful biomarker for HD. And this is so crucial because it's actually the first piece of research that computationally analyzes biomedical signals for Huntington's disease in this way. And if you would like to read the full paper, of course, it's published on arXiv currently, and its consideration at other journals. I'd like to conclude this presentation by thanking some important people. I'd like to thank my mentor, Christine Yoon, for all of her mentorship and support. I'd like to thank the Polygence staff for organizing this excellent program. I'd like to thank Emily Nguyen from USC for advice on paper writing. I'd like to thank Nick from Polygence for being an excellent admissions advisor. I would also like to thank my parents for all of their financial, moral, and loving support. Thank you so much.
Great job, Sucheer. Really clear, awesome presentation. We've got some Q&As. The first one is, hi, Sucheer. I'm curious, how did you access your data?
Sure. So if I had to search quite a bit online, since this isn't something that had data for, but I looked at university repositories. One of them was this data set from Lancaster University in the UK, and they essentially made this data. And I talked with them and had to gain permission to actually use the data set. But after I did that, I was able to gain access with the data. And I also did some formatting to convert them all into CSV files to, get them ready for the whole project.
Awesome. Jin asked as well, how did you decide to pick Huntington's versus other neurodegenerative diseases like Alzheimer's, ALS?
Great question. So biomedical signals have already been applied to many diseases such as schizophrenia, Alzheimer's, Parkinson's. I'm not 100% sure about ALS. Maybe that's something for me to consider, but it's been applied to many neurodegenerative diseases already, and it's seen success. However, it has not been applied to Huntington's. So I wanted to do something that hasn't been done before and could be useful. So that's why I did Huntington's disease.
That question kind of answers one that was raised by one of your reviewers too. That was, are these signals used for diagnoses or monitoring of other diseases already?
Yes.
Or how do you feel this could expand to other diseases?
Yeah, so they're already used for some diseases. And the reason I picked Huntington's again was because, it's also a neurodegenerative disease. So I thought, hey, maybe we could also see abnormalities here, and we could. And it could maybe be expanded to other diseases, although as we saw, EEG was the most important signal. So unless it's a neurological disease, it might be a little bit difficult to accurately predict the disease.